Good morning, dear learners. In our today's class, we are going to talk about the techniques of immunodiffusion, and then we are going to have the short discussion on the technique of immunoelectrophoresis. So first of all, we should know that what is the meaning of immunodiffusion. So basically, we know that what is the meaning of diffusion. Diffusion is when a substance is moving from the region of its high concentration to the region where its low concentration is present. That is what we call diffusion. And when we add the prefix immuno to this word, it becomes immunodiffusion. It simply means that the antigens and antibodies now will move from the region of their higher concentration to the region of their lower concentration. And surprisingly, all of this takes place on a plate or on a slide. So in the case, let us assume that I have a slide. Now on the slide I will pour what is popularly called as the agar gel. So this is my agar gel which I have poured on the slide. Wait. So here is my agar gel. This is the agar gel on my slide. Now why we are adding agar gel? Because agar gel will provide a particular scaffold or will provide a pathway through which the molecules will diffuse. Then what do? I will make a well here on the agar gel and another well here. In one of the wells I will coat and I will add the antigen, and in the another wells I will coat the particular antibody. So let us assume that the antigen I have loaded in the red color. This is my red colored antigen, and this is the serum or a particular solution which is rich in antibodies, which is very specific to that antigen. So if it is antigen X, so it is anti-X antibody, and this will be the antigen X. Now what happens? As I told you that these will start diffusing, so the diffusion takes place. Now we already know that this is the region where the concentration of antigen is highest, and this is the region where the concentration of antibody is highest. Now they have all the directions where they can move, but here they are limited by the boundaries, so they cannot move further here because then there is the edge. So one direction where they are free to move is this way. So the antigens start moving this way and the antibodies start moving this way. So I can even draw it with the lines. Suppose this is my antigen, it is moving here in this way. This is the direction of moving of antigen, and similarly this is the direction of moving of antibodies. In its most raw form, this is only what immunodiffusion is. So antigens have diffused and antibodies have also diffused. Now there will be a particular place where they will meet, and the place where they will meet, there the precipitation reaction takes place. So suppose if this is the place here, this is the place where they are meeting. So in this area or in this region the precipitation reaction takes place. So here I can say that there is a reaction which we call as precipitation. So here I have the precipitation reaction wherein the antigen and the antibody will combine with each other. If you know the molecular aspects, like if this is the green colored antibody, so this is the red colored antigen, and this way they are attached to each other. So this reaction takes place here and the precipitation occurs at this place, and here we will get a colored band. Not a colored band, it can be a precipitate, so let us say it is of this only, but a bit of dark colored band. So let us assume that this is the place where that reaction has taken place. So this is the zone where the precipitation has taken place, and this zone is also referred to as, this zone is called a zone of equivalence. Now this zone of equivalence and these regions can be plotted on the graph as well. So if I try to plot them on the graph, suppose this is my x-axis, this is my y-axis, and this is also my y-axis. This can be plotted in a simple terms like this. Okay, this is perfect. Let me make it a bit like that way. Okay, this is perfect. So this is the zone of equivalence here. So what I say is that this place, this is the place where I have the zone of equivalence. These are the regions where I have antigens in excess, and these are the regions where I have the antibodies in excess. This is what we plot generally when we have to talk about the immunodiffusion. Now this whole technique which I have talked about is what we call as the diffusion of the antigens and antibodies, or the immunodiffusion and the precipitation. Now I will talk about that why does the precipitation happens. So see, when the antigen and antibodies link together, like as I was considering this example here, so this can be in multiple forms, right? So when it is in the multiple forms, suppose one is here, then one is again here, then one is again here. Let's see if I can turn it. No, I cannot turn it, but you can understand that yes, this is also the place. Suppose then one comes out here, one here, one here. These are the antigens only, and then these are the antibodies. So what these antibodies are doing is, actually these antibodies are cross linking all of these antigens, and this, if you particularly observe, becomes a kind of lattice. This is a kind of lattice which has become here. So I will highlight this lattice here. So this is the place where a kind of lattice has formed, where multiple antigens are interacting with each other and they have settled down because they have precipitated. So this is the place where lattice formation takes place. So we say that multiple antigens are linked together by the antibodies to form the lattice. Now if I focus on this area, just focus specifically on this area, particularly this area. It has antigens in high amount, but the antibodies available are low. So the lattice formation does not accurately take place. Here also the antibodies are very high in number, but the antigens available are less. So here also the lattice formation will not take place. The lattice formation only takes place at the zone of equivalence, because we have there the optimal concentration of the antigens and the antibodies. So this is very important concept and it can be graphically plotted. Now, on the basis of what we have learned up till now, we can divide this immunodiffusion methods into two broad categories. So if I talk about the immunodiffusion, so these techniques of immunodiffusions can be two types. One of them is what we call as Ouchterlony double diffusion. It is merely a qualitative assay. Means you have to just detect the presence of whether the antigen is present in the serum or not, or antibody is present in the serum or not. Secondly, we have the Mancini's method. Here this is the Mancini's method, which is more of the quantitative assay. Here we can even determine the quantity of the particular substance in our serum, whether be it antigen or be it antibody. Now we have to discuss the two of them in details, but the Ouchterlony method, 3/4 of the Ouchterlony method I have already discussed above, wherein if the precipitation line forms and if the zone of equivalence comes, this is what we call as the that the antigen is definitely present, and because of that only the antibody has reacted to that antigen. So this is a confirmatory experiment, or it is a qualitative assay. Another form of the same Ouchterlony method relies on the circular plates rather than a glass slide. So there what do we have is, we particularly have a kind of, let's say this is the kind of circular plate which we have. Okay, this is the circular plate which we are having. Now what do we do? Please kindly listen to it very carefully. Again, On the whole of the plate, that is very fixed, that on the whole of the plate we will be having the agar poured. Now in the protocols you have to learn that how much of the agar is poured and all. That is what one can learn, but the concept should be very clear. So this is the pouring of agar which has done, and approximately 5 ml of agar we are pouring. So this is agar which we have poured. Now what do? We will carve out the specific holes. So let us say the 2 holes we have carved out here, and then 1 hole we have carved out here. Now these specific holes are for antigens, and this specific hole is for antibody. Now let me make it in duplicates because I will require it. Let me make it a bit smaller. Okay, this is perfect. Now we have to discuss each of them one by one in detail. Let's move on to the first one here. The first one here, the Ouchterlony method only. Here what we are doing is, in the green we are pouring the particular antibody, and here we are pouring the particular antigen. Okay. Now suppose if the two antigens are similar, we have to detect whether the antigen which we were confirming, is it even that antigen or not. So here what will happen is, this is the well of antigen, this is the well of antigen 1, this is the well of antigen 2, and this is the well of antibody. Now suppose the two antigens, let us assume the two antigens are very similar to each other. When the two antigens become very similar to each other, what happens is that now the antibody will start diffusing from its place, and the antigens will also start diffusing from the place. Now there will be a point where the zone of equivalence will be reached, and the zone of equivalence would be in a shape of a perfect curve. This would be the zone of equivalence. So here what can we observe is that here we had this antigen, we had this antigen, they started diffusing, and here also it started diffusing, and here the antibody started diffusing, because the two antigens are very similar. What do you mean by similarity? In similarity we means the epitopes of the two antigens alike. So we can say that they are isotypic in nature. So here what happens is that we particularly get this as a line, which is what we call as the zone of equivalence, and this is in a form of perfect curve. Now what happens in this case is that the two antigens are very identical, so we call it as identity immunodiffusion. Now let's take another case. We have kept this antigen here only, that is what we have artificially kept, and then we are assuming that it would be the antigen in the patient would also be similar, but it is not. Let us assume it is not. Then what happens? So this is our antigen 1 this time, and this is our antigen 2, and antigen 1 and antigen 2 are not at all similar to each other. They are isotypically very different, and this is the antibody. Now again the same thing starts happening. The antigen 1 will start to diffuse, antigen 2 will start to diffuse, and antibodies will also start to diffuse in the agar. But this time the kind of curve which we get is very surprising. The kind of curve is, we get this particular, this shape of curve, that is the X shape of curve. Whenever we get this X shape of curve, we say that the two antigens are not at all alike to each other, their epitopes are very different, and hence the antibody has reacted differently to them, and this is what we call as non-identity immunodiffusion. The third one is where partially the epitopes are matching and partially they are not matching. So how do we write it? We write it as partial identity immunodiffusion, or we can also write partial entity Ouchterlony immunodiffusion, whatever you write. So let's say some part of it was red and some part of it was also red. Then the remaining part, let's say here it was pink colored, and here it was, let's assume, blue colored. But there was a partial point where we say that the antigen 1 and the antigen 2 had the similar kind of isotopes. This is where we have loaded the antibody, which is always of green color only, let us assume. Now, what kind of curve here I get, or what kind of equivalence zone I get here, is like this. This is the kind of equivalence zone which I perfectly get here. And this is what we call as the partial identity immunodiffusion. So these three kind of immunodiffusions can come, and if any one of them, if you are aware of the kind of curve which we get from any one of them, so you will be also very sure that what kind of immunodiffusion is that. So this is the whole concept of Ouchterlony immunodiffusion. There is another kind of immunodiffusion which we should not study in much details, but yes, at least for the knowledge we should know. The second kind of immunodiffusion is Mancini's method. Mancini's method. Now what do we do in Mancini's method? Basically, we are taking the plate again. So let us say this is the plate. But in this plate we are doing one difference. The agar which we were adding now this time also contain the particular antibody. Means antibody and agar's mixture we are adding. So let's say because of the addition of antibody, the color of agar changed to this. So what do I say and what do I refer here is that the antibody is uniformly spread in whole of the plate. Okay. Now what do we need to do? We need to let us duplicate it. Now here in the Mancini's method, one of them will be control and one of them will be test. Now here we are going to, let's assume we take the four holes here. In this I'm loading the antigens. Here suppose I have less concentration of antigen, here more concentration, here more concentration, and here more concentration. So more the concentration of antigen, more far will be the let's say the zone of equivalence circles we will get. See, what I mean to say is the antigen, what will the antigen do now in the well? It will start diffusing out. So it will diffuse here also, but here it will diffuse out more because the concentration of that antigen is more. So the lines which we are going to get is very important to know. Here let's say we are getting the line here. Now here the concentration is increased, so the zone of equivalence got far. Here the concentration increased further, so zone of equivalence got even far, and here it got the farthest. So as we move on from here to here, the concentration increased, so the equivalence zone or the circle or the diameter increased. Here it increased more and here it reached to its maximum peak. Now here what do? We will coat the wells again, or we will make the wells, and we will add the antigen here whose concentration is unknown. Now suppose in these wells, because I have added a similar antigen, so I will definitely get the similar kind of concentration. So suppose here this is what I have got. Now how do I draw this? The similar kind of concentration I'm getting, suppose here also I got, here also I got, and here also I got. Now I can easily compare it with this one, and I can say that what or where do my concentration of antigen lie, in which range. So it is more or less a comparative kind of immunodiffusion. That is what Mancini has deduced, and this is very important because it is a quantitative immunodiffusion, whereas Ouchterlony is only the quantitative immunodiffusion. And there is a major drawback of these immunodiffusions, that is it will require the higher concentration and the higher amounts of antigen and antibody, means it is not that sensitive. So this is what we need to learn about these methods. Let us move to the last method, a short note on the immunoelectrophoresis. What is immunoelectrophoresis? Okay, so I'm writing here. The third is the third one being is immunoelectrophoresis. Again, the similar kind of thing will be applied here also. This we have is our glass slide. Here we have coated it with agar, obviously. This is my agar coating. Wait, this is my agar on it. Now what I do is, now this is very important, we have to listen it very carefully. Here I will make a well in the center, and here what do I do is I will then add my particular serum. So here what is the difference? Here I am adding the serum here, and I am unaware that what